evolution doesn't have a memory, but it keeps telling the same story. Right now, somewhere in the ocean, a crab is being born. Not from crab parents necessarily, but from something that wasn't quite a crab until evolution bent it into one. This has happened at least 5 separate times. 5 different lineages separated by millions of years and entire oceans, independently stumbling onto the same body plan. The same flat shell, the same tucked-in tail, the same sideways scuttle. Scientists call this carcinization, but it's just one example of something far stranger happening across all life on Earth. Wings evolved 4 separate times, camera eyes twice, venom dozens of times. The ability to fly, to see, to photosynthesize, to give live birth. Life keeps solving the same problems with the same solutions, as if reading from a script it's never seen before. It's like watching different composers, centuries apart, never having heard each other's work, all writing the same symphony, note for note, movement by movement, each believing they've discovered something new. But here's what keeps evolutionary biologists awake at night. If evolution is random, if mutations are chance, if natural selection has no foresight, then why does life keep arriving at the same destinations? Why does the universe of possible forms keep collapsing into the same handful of answers? What we're about to uncover suggests that life isn't as free as we thought, that beneath the chaos of mutation and the randomness of chance, there are deeper rules. Constraint fields that channel evolution, like water, finding the same valleys again and again, regardless of where the rain begins. This is Omni, stories about life, matter, and the mystery in between. Like the video, drop your thoughts in the comments, and subscribe if you believe curiosity should never end. Picture 2 eyes staring at each other across 600 million years. One belongs to you, the other to an octopus drifting in the dark waters off the coast of Japan. Your eye is reading these words. The octopus's eye is tracking the shadow of a crab moving across the seafloor. Both eyes have a cornea, an iris, a lens. Both have a retina lining the back, converting photons into electrical signals. Both can focus, adjust for light, perceive movement with extraordinary precision. You're looking at convergent evolution's masterpiece. 2 species that last shared a common ancestor before brains existed, before bones existed, before anything resembling a sophisticated body plan existed. Both independently evolved nearly identical solutions to the problem of sight. The last creature you both descended from was probably something like a flatworm, eyeless, simple, crawling through Precambrian mud. From that humble origin, your lineage went one direction, toward a backbone, toward limbs, toward primate ancestors swinging through ancient forests. The octopus's lineage went somewhere completely different, toward a soft body, toward 8 arms, toward a life of jet propulsion through open water. Nothing in your shared history explains why you both ended up with camera eyes. Nothing in the laws of natural selection required it. Plenty of animals see perfectly well with compound eyes, like insects, or mirror eyes, like scallops, or pinhole eyes, like nautiluses. The octopus could have evolved any of these. So could you. But you didn't. Both lineages, working with completely different genetic toolkits, building eyes from completely different embryonic tissues, using completely different developmental pathways, arrived at the same design. As if the universe of possible eyes narrows down to a few optimal solutions, and evolution keeps finding them. According to research published in 2004 by Atsushi Ogura and colleagues at the National Institute of Genetics in Japan, when they compared the genes expressed in octopus eyes to those expressed in human eyes, they found something remarkable. 729 genes were commonly expressed between the 2 species. That's 69.3% overlap. Not because of shared ancestry, but because both species, independently, recruited the same ancient genetic toolkit to build their eyes. The genes existed long before either species did. They were sitting there in the genome of that eyeless flatworm ancestor, doing other jobs, maybe building body segments, or controlling cell division, or managing embryonic development. Then, twice, separated by hundreds of millions of years, Evolution reached into that same genetic library and said, these ones, these genes right here, let's use them to build an eye. It's like two architects in different centuries never having met, both walking into the same ancient warehouse full of building materials and independently deciding to construct the exact same cathedral, using the same stones, the same arches, the same proportions. But eyes are just the beginning. Crabs, as we mentioned, have evolved 5 separate times from non-crab ancestors. According to a 2021 study by Joanna Wolf and her team at Harvard University published in Bioessays, this process, called carcinization, happens so reliably that paleontologists can watch it unfold in the fossil record. Different families of crustaceans, in different geological periods, in different oceans, all independently deciding that the optimal body plan involves a flat, round shell and a tail tucked underneath. King crabs evolved from hermit crabs, porcelain crabs evolved from squat lobsters, even some true crabs evolved from different lineages of true crabs, as if life keeps returning to the same form because the form itself is inevitable. Wings tell the same story. Insects evolved wings 350 million years ago. Pterosaurs evolved them 230 million years ago. Birds evolved them 150 million years ago. Bats evolved them 50 million years ago. 4 completely separate inventions, 4 completely different starting anatomies. Insect wings are outgrowths of the body wall. Pterosaur wings are elongated fourth fingers. Bird wings are modified forelimbs with feathers. Bat wings are stretched skin between extended finger bones. None of them could have copied each other. None of them inherited wings from a common ancestor. Yet all of them arrived at powered flight using the same fundamental principles: a lightweight frame, an airfoil surface, and the ability to generate lift through motion. Venom evolved independently over 60 times. Joanna Wolf at Harvard estimates that placental and marsupial mammals have evolved remarkably similar body plans despite evolving on separate continents. C4 photosynthesis, a more efficient way of capturing carbon dioxide, evolved independently at least 45 times in different plant families, according to research from the University of Wisconsin-Madison published in 2024. It's everywhere. Once you start looking, you can't stop seeing it. Life keeps writing the same chapters in different books. The question that keeps echoing is, why? If evolution is a process driven by random mutation and natural selection, if there's no plan, no blueprint, no predetermined destination, then why do these patterns keep emerging? Why does the space of all possible forms seem to have attractors, like gravity wells that pull different lineages toward the same solutions regardless of where they start? The answer changes everything we thought we knew about how free evolution really is, about how many ways there are to solve the fundamental problems of being alive, about whether the development of complexity, intelligence, even consciousness itself, is random chance or something closer to inevitability. Tonight, we're going deep into convergent evolution. We'll trace how life keeps reinventing the same tools, from molecular machines to body plans to behaviours. We'll explore the mathematical and physical constraints that seem to channel evolution into preferred pathways. We'll ask whether the tape of life, if rewound and played again, would produce the same results. And we'll confront the possibility that the universe of biological forms isn't infinite at all, but surprisingly small, a limited set of viable solutions that evolution discovers over and over again. Because understanding convergent evolution means understanding the hidden grammar of life itself, the rules we didn't write but can't break, the solutions that work so well that nature keeps going back to them, as if reading from a book that exists nowhere but everywhere at once. Let's begin where all patterns begin, with the question of whether they're accidents or inevitabilities. The hardest part about understanding convergent evolution is accepting what it implies about freedom. We like to think evolution is open-ended, random mutations, natural selection, endless possibilities. If you could rewind the tape of life and let it play again, most people assume you'd get something completely different. Maybe intelligence never evolves, maybe mammals never appear. Maybe life stays aquatic forever, or never leaves the bacterial stage at all. But convergent evolution whispers something else. It suggests that the tape, no matter how many times you rewind it, keeps playing the same scenes. Different actors, same script, different timeline, same ending. Let's start with the crab problem, because it's almost absurd enough to make you question whether randomness is even involved. Carcinization is the scientific term for evolving into a crab. The word was coined in 1916 by English zoologist Lancelot Alexander Borradaile, who noticed what he called the many attempts of nature to evolve a crab. He was being poetic, but he was also being accurate. Nature has attempted to evolve a crab at least 5 separate times, and possibly more. According to Joanna Wolf's 2021 study published in Bioessays, the crab body plan has independently evolved in king crabs, porcelain crabs, hairy stone crabs, sponge crabs, and most eubrocurans, which include the vast majority of what we think of as true crabs. Each of these lineages started from a non-crab ancestor, something more lobster-like or shrimp-like, with a long body and an extended tail. Then, over millions of years, each one independently underwent the same transformation. The body flattened, the shell widened, the tail tucked under, the legs spread to the sides, optimized for scuttling sideways rather than swimming forward. What makes this remarkable isn't just that it happened 5 times, it's that it happened within a relatively narrow branch of the tree of life. All 5 carcinizations occurred within decapod crustaceans, a group that also includes lobsters, shrimp, and crayfish. These aren't wildly distant lineages, they're cousins, evolutionarily speaking, and yet give them enough time and the right environmental pressures, and they all solve the problem of survival the same way: become a crab. King crabs are perhaps the most striking example. They evolved from hermit crabs, those soft-bodied creatures that protect themselves by living inside discarded snail shells. Hermit crabs have asymmetrical, spiraled abdomens designed to fit into spiral shells. When king crabs evolved, they abandoned the shells entirely and developed hard exoskeletons. But here's the strange part. Their abdomens are still asymmetrical. They still bear the marks of their shell-dwelling ancestry, even though they no longer need shells. Evolution dragged them toward crabness so powerfully that they gave up their defensive strategy and evolved armor instead, all while retaining the twisted body plan of their ancestors. Why does this keep happening? Wolf and her colleagues suggest several possibilities. The tucked tail reduces vulnerable flesh exposed to predators. The flat, round shell might help with scuttling sideways, which could be more effective for fitting into crevices or navigating complex terrain like rocky shores or coral reefs. The body plan might simply be mechanically efficient for the kinds of environments these creatures inhabit. But here's what should bother you. If the crab form is so advantageous, why didn't it evolve everywhere? Why don't fish become crabs? Why don't insects become crabs? The answer reveals something profound about the constraints operating beneath the surface of evolution. Crabs keep evolving within decapods because decapods have the right building blocks. Their body segments, their limb structures, their developmental programs all make the crab form accessible. Evolution can reach it from where they start. For a fish to become a crab would require rewriting so much fundamental biology that natural selection would never get there. The path is too long, too twisted, too many intermediate steps would produce non-viable organisms, so fish never become crabs. But within decapods, the path to crabness is short enough, viable enough, advantageous enough that evolution keeps finding it. This is the beginning of the real story. Evolution isn't exploring all possible forms, it's exploring a constrained space, and that space has preferred regions, optimal solutions that keep getting discovered regardless of the starting point. Think of it like this. Imagine you're hiking in a landscape where valleys represent stable, advantageous body plans. You start at different points on different mountains, but every time a rainstorm happens, water flows downhill, and every stream, no matter where it starts, ends up in the same valleys. Not because the water is intelligent, not because it has a goal, but because the landscape itself channels flow towards certain destinations. The valleys are inevitable, given the terrain. Morphospace is the term biologists use for this landscape. It's the mathematical space of all possible body forms an organism could theoretically have. Each point in morphospace represents a different configuration: longer legs, shorter tail, wider body, different number of segments. Most of morphospace is unoccupied. Most possible forms never appear in nature, not because they're impossible to reach, but because they're not stable, not functional, not advantageous enough for natural selection to preserve them. According to research from the University of Vienna published in 2024, morphospace exploration reveals that developmental constraints, the rules governing how embryos grow and differentiate, create forbidden zones where certain body plans simply cannot be generated. You might imagine a creature with 7 legs, but if the genetic toolkit that builds legs operates in pairs controlled by bilateral symmetry, genes, 7-legged animals become inaccessible. Not because they'd be bad at survival, but because development can't produce them in the first place. The crabs occupy a region of morphospace that's both accessible from multiple starting points within decapods and functionally advantageous for the environments they inhabit. Evolution keeps flowing toward that valley because the valley exists, stable and deep, carved into the landscape of possibility by physics, chemistry, and developmental biology. But crabs are just one example. Once you start looking, the pattern appears everywhere. Camera eyes evolved twice, once in vertebrates, once in cephalopods like octopuses and squids. Atsushi Ogura's research published in Genome Research in 2004 found that despite 600 million years of separate evolution, 69.3% of genes expressed in octopus eyes are also expressed in human eyes. These aren't inherited from a common ancestor with eyes. The common ancestor of vertebrates and cephalopods was probably something like a simple flatworm with basic light-sensitive patches at best. Instead, both lineages independently recruited the same ancient genetic toolkit to build their eyes. The genes were already there doing other jobs. PAC6, the master control gene that orchestrates eye development in both lineages, existed in that ancient common ancestor. It wasn't building eyes then because eyes didn't exist yet. It was probably involved in basic body patterning or neural development. But the gene had properties, binding sites, regulatory capabilities that made it useful for controlling the complex cascade of events required to build an eye. So when eyes started evolving independently in both lineages reached for PAC6. It's like 2 engineers in different countries, building bridges independently, both reaching for steel. Not because they copied each other, but because steel has properties that make it ideal for bridge building. The solution isn't infinite. The properties of materials constrain what works. Wings tell the same story with even more repetition. Insects evolved wings about 350 million years ago. Pterosaurs, those flying reptiles that dominated Mesozoic skies, evolved wings around 230 million years ago. Birds evolved wings about 150 million years ago. Bats evolved wings roughly 50 million years ago. 4 independent inventions, 4 completely different anatomical starting points. Insect wings are outgrowths of the body wall, extensions of the exoskeleton with no internal bones. Pterosaur wings were skin membranes stretched between massively elongated 4th fingers. Bird wings are modified forelimbs covered in feathers, with the feathers doing most of the aerodynamic work. Bat wings are also modified forelimbs, but the membrane stretches between 4 elongated fingers instead of 1, yet all 4 produce powered flight using the same aerodynamic principles. All 4 generate lift by moving an airfoil surface through air. All 4 had to just solve the same problems: lightweight construction, sufficient wing area, muscle attachment points that allow flapping, mechanisms for controlling pitch and yaw. The physics of flight doesn't care about your ancestry. Air is air, gravity is gravity, lift is lift. If you want to fly under your own power at animal scale, you need a wing. And a wing has certain requirements that narrow the space of viable solutions dramatically. Evolution keeps finding those solutions because they're the only ones that work. This is the pattern. This is what convergent evolution keeps revealing. The space of possible solutions to biological problems is smaller than we thought. Much smaller. And here's where things get philosophically uncomfortable. Stephen Jay Gould, the famous evolutionary biologist and paleontologist, argued in his 1989 book Wonderful Life that if you could rewind the tape of life and let it play again from the Cambrian explosion, you'd get something completely different. Different body plans, different dominant lineages, maybe no vertebrates at all, maybe no intelligence. The outcome he claimed would be unpredictable because evolution is fundamentally contingent, dependent on accidents, chance events, lucky breaks that could easily have gone another way. Replay the tape, get a different movie. Simon Conway Morris, Gould's colleague and a paleontologist at Cambridge University, spent years looking at the same fossils Gould studied and came to the opposite conclusion. In his 2003 book Life's Solution: Inevitable Humans in a Lonely Universe, Conway Morris argued that convergent evolution reveals something profound. The tape of life, if replayed, would produce strikingly similar results. Not identical, but convergent. Similar eyes, similar wings, similar intelligence, similar solutions to the same problems. Conway Morris famously stated, "The evolutionary routes are many, but the destinations are limited." This isn't a fringe position anymore. Recent research increasingly supports the idea that evolution, while exploratory and unpredictable in its details, is constrained and directional in its outcomes. The space of possible solutions is smaller than the space of possible mutations. Much smaller. Take echolocation. Bats evolved it. Toothed whales evolved it. Some birds, like oilbirds and cave swiftlets, evolved it. Even some shrews and tenrecs evolved rudimentary versions, at least 4, possibly 6 or more independent origins of the same biological sonar system. Each lineage started with different anatomy, different genes, different ecological pressures, but all of them converged on the same solution. Emit sound, listen for echoes, build a spatial map from the timing and frequency of returns, and it's not just behavioural convergence. According to research published in 2010 by Stephen Rossiter and his team at Queen Mary University of London, the genetic changes underlying echolocation in bats and dolphins show molecular convergence. Mutations in are the same genes, changes to the same proteins. Both lineages independently modified their Preston gene, which affects the outer hair cells in the cochlea, making them more sensitive to high-frequency sounds. Natural selection, working independently on different genomes, found the same genetic solution. This suggests that the space of genetic solutions to echolocation is even narrower than we thought. Not just any random mutation can produce sonar. There are specific molecular pathways that work, specific protein modifications that enhance high-frequency hearing, and evolution keeps finding them because they're the ones that actually function. The same pattern appears in electric fish. In South America, knifefish evolved the ability to generate and sense electric fields for navigation and communication in murky water. In Africa, completely unrelated elephant fish evolved the same ability. 2 independent origins, 2 different continents, 2 separate evolutionary trajectories. But both use specialized electric organs derived from modified muscle tissue. Both generate weak electric fields. Both detect distortions in those fields caused by nearby objects. C4 photosynthesis, a more efficient way of capturing carbon dioxide that evolved in hot, dry environments, has arisen independently at least 45 times in different plant lineages, according to research from the University of Wisconsin-Madison in 2024. That's not a typo. 45 times. The solution is complicated. It requires anatomical changes to leaf structure, biochemical modifications to carbon fixation pathways, and coordination between different cell types. Yet evolution keeps assembling it from scratch, because in hot, dry conditions where photorespiration becomes a problem, C4 photosynthesis is simply better. Venom has evolved over 60 times across the animal kingdom. Snakes, spiders, scorpions, cone snails, jellyfish, platypuses, shrews, slow lorises, all independently discovered that injecting toxic proteins into prey or predators is an effective strategy. The specific toxins differ. Snake venom and spider venom work through different molecular mechanisms, but the basic solution, specialized glands, delivery systems, proteins that disrupt physiology, keeps emerging. Even complex social behaviors converge. Eusociality, where individuals in a colony divide into reproductive and non-reproductive castes and cooperate to raise offspring, has evolved independently in ants, bees, wasps, termites, naked mole rats, and certain shrimp. The details differ. Termites have kings and queens, bees have only queens, naked mole rats organize differently. But the underlying pattern is the same: sacrifice individual reproduction for collective success. This is what Conway Morris means by attractors of functionality. Certain solutions work so well, and the pathways to reach them are accessible enough, that evolution keeps flowing toward them like water finding the same valleys. The landscape of biological possibility isn't flat. It has peaks and valleys, optimal regions and impossible zones, and those features of the landscape constrain where evolution can go. But why? What creates these constraints? Physics is one answer. Flight requires generating lift, and lift has specific aerodynamic requirements. Wings need a certain shape, a certain surface area relative to body mass, a certain structural strength. You can't violate the laws of fluid dynamics no matter how creative your mutations are. So wings converge on functional forms that work within those physical constraints. Chemistry is another. Vision requires molecules that change shape when hit by photons. There are only so many molecular structures that do this reliably. Opsins, the proteins that underlie vision across nearly all animals, share deep chemical similarities because chemistry itself limits the options. You need a protein that can hold a chromophore, that can change conformation rapidly when the chromophore absorbs light, that can trigger a signaling cascade in response. The solution space is narrow. Development creates its own constraints. According to a 2024 study from the University of Vienna using computational models of embryonic development, certain body plans are simply easier to generate than others, given the mechanics of how cells divide, move, and differentiate. If early development operates through certain conserved signaling pathways, and it does, with Hox genes and WNT pathways, and notch signaling appearing across vast stretches of the animal kingdom, then only certain morphologies are developmentally accessible. Imagine trying to build a house using only the tools in your grandfather's garage. You might want to build a geodesic dome, but if the only tools you have are a hammer, saw, and level, you're probably going to end up with something more traditional. Not because traditional houses are objectively better, but because they're what those tools can easily produce. Evolution's toolkit, the genes, proteins, and developmental programs inherited from ancestors, constrains what structures can be built. This is why you don't see wheels in biology, even though wheels are mechanically efficient for movement on flat surfaces. Wheels require parts that rotate continuously and independently from the body, which means no permanent connections for blood vessels or nerves. Development can't easily produce a structure that's both part of the organism and completely free to rotate. So legs, fins, wings, and jet propulsion all evolved, but wheels didn't. Not because wheels wouldn't work, but because they're developmentally inaccessible given how animal bodies are built. The constraints run deeper than individual features. Entire body plans seem to have preferred forms. Bilateral symmetry, for instance, appears in almost all mobile animals because moving through space with a front and back, a left and right, is mechanically advantageous. Radial symmetry works for sessile or slow-moving creatures like sea anemones and jellyfish. But once you start actively pursuing food or fleeing predators, bilateral symmetry becomes almost inevitable. Segmentation, repeating body units along the anterior-posterior axis, evolved independently in arthropods, annelids, and chordates, three completely separate inventions of the same architectural principle. Why? Because segmentation allows for modularity, specialization, and redundancy. Lose one segment, the others can compensate. Specialize different segments for different functions. Locomotion, feeding, sensing, reproduction. It's an architecture that works. Even at the molecular level, convergence appears. Antifreeze proteins, which prevent ice crystals from forming in the blood of cold water fish, have evolved independently at least 5 times in different fish lineages. The proteins have different sequences, different structures, but they all work by the same mechanism, binding to the surface of tiny ice crystals and preventing them from growing. Chemistry provides only so many ways to solve this problem, and evolution keeps rediscovering them. The pattern is unmistakable. Whether you're looking at molecules, cells, organs, body plans, or behaviors, evolution keeps arriving at similar solutions. Not because those solutions are copied, but because the underlying constraints, physical, chemical, developmental, channel evolution towards specific outcomes. This brings us back to the question that haunted Gould and Conway Morris. If evolution were replayed, what would happen? Gould's argument rested on contingency. Evolution, he believed, is path-dependent. Small accidents early in history cascade through time, creating outcomes that couldn't be predicted from first principles. If a slightly different mutation happens in a Cambrian worm, if a different lineage survives a mass extinction, if one population gets isolated on a different island, the entire trajectory of life changes. Replay the tape, get a different movie every time, Conway-Morris looked at the same evidence and saw convergence everywhere. The same eyes, the same wings, the same solutions appearing again and again across vast stretches of evolutionary time and phylogenetic space. To him, this suggested that evolution isn't exploring infinite possibilities. It's exploring a constrained landscape with preferred destinations. Replay the tape, and you'd get similar outcomes because the underlying physics, chemistry, and developmental biology channel evolution toward the same solutions. The evidence increasingly supports Conway-Morris. But with a crucial caveat: the constraints aren't absolute, and the outcomes aren't predetermined. Evolution is constrained exploration. It has preferred directions, but within those directions, tremendous variation remains possible. Think of it like language. All human languages share deep structural similarities: nouns and verbs, subjects and objects, ways of marking, tense and number and negation. These similarities exist despite languages evolving independently across the globe with no contact between populations for tens of thousands of years. The constraints come from how the human brain processes information, from the problems language needs to solve, from the physics of speech production. But within those constraints, we get thousands of distinct languages, each with unique vocabulary, grammar, and structure. Evolution works the same way. The space of viable body plans is constrained by physics, chemistry, and development. But within that constrained space, immense diversity remains possible. Consider body plans themselves. There are roughly 35 recognized animal phyla, representing the major architectural types of animal bodies. Why only 35? You can imagine other configurations. Bodies with odd numbers of segments, radial symmetry combined with bilateral features, novel organ arrangements. Science fiction writers invent them constantly. Yet in 600 million years of animal evolution, only 35 basic body plans have persisted. According to developmental biology research, the answer lies in what's called the phylotypic stage, a conserved period during mid-embryonic development when body plans become fixed. Research published in Nature Ecology and Evolution in 2017 found that mid-embryogenesis shows remarkably low variation across species compared to early and late developmental stages. This is the developmental hourglass: lots of variation at the beginning, a constrained bottleneck in the middle where the basic body plan gets established, then increasing variation again as specific organs and features develop. During this phylotypic stage, multiple developmental processes must coordinate simultaneously. The neural tube is forming, somates are segmenting, major organ systems are being specified. Any major disruption during this period tends to be lethal, because so many interconnected processes are happening at once. This creates a conservation trap. Once a lineage passes through a particular phylotypic stage, it's locked into that body plan. Vertebrates will always have a notochord, bilateral symmetry, a dorsal neural tube, pharyngeal slits. Modifications can happen before the phylotypic stage, adaptations can occur after, but the core architecture is fixed. This is why you don't see hybrid body plans. No animal is half arthropod, half chordate. The developmental programs are incompatible. You can't mix and match body plans because development is a cascade of integrated processes, and once you commit to one pathway, switching becomes impossible without destroying the entire developmental sequence. The constraints get even more specific. Within vertebrates, tetrapods, animals with 4 limbs, all share a remarkably similar limb structure. 1 bone in the upper limb, 2 in the lower limb, then wrist or ankle bones, then digits. Whales have it, bats have it, humans have it, even, though our limbs do completely different things. Whale flippers evolved from walking legs, but they retained the underlying skeletal pattern, because modifying that pattern requires rewriting the genetic regulatory networks that control limb development, networks that are deeply integrated with other aspects of body patterning. Research published in 2018 examining developmental constraints on genome evolution found that genes expressed during the phylotypic stage are under stronger purifying selection than genes expressed at other times. They're older genes, more conserved across species, less likely to be duplicated or lost. The genome itself reflects the constraints of development. Mess with these genes, and you destroy the developmental program. Natural selection ruthlessly eliminates such changes, but constraints don't just limit, they also enable. By fixing certain aspects of body plans, constraints create platforms for variation in other dimensions. Once bilateral symmetry is established, evolution can explore variations in limb number, body segment specialization, sensory organ placement. The constraint channels exploration into productive directions rather than letting it wander randomly through impossible configurations. Imagine trying to improvise music. If you know no music theory and no instrument, you're free to make any sound, but most of those sounds won't be musical. Learn a scale, learn chord progressions, learn rhythm, and suddenly you're constrained. You can't play just any note at any time, but within those constraints you can create infinite melodies, harmonies, compositions. The constraints aren't limitations, they're the structure that makes meaningful variation possible. Evolution works this way. Developmental constraints, physical constraints, chemical constraints, they all narrow the space of exploration. But they narrow it toward regions that actually work, toward solutions that can be built, maintained, and improved. And within those regions, vast variation remains. This explains both the repeatability of convergence and the uniqueness of individual lineages. Eyes keep evolving because eyes solve the problem of converting light into spatial information, and there are only so many ways to do that given the physics of light and the chemistry of photoreceptor molecules. But each type of eye has unique features: compound eyes in insects, camera eyes in vertebrates and cephalopods, mirror eyes in scallops, pinhole eyes in nautiluses. The solution space is narrow enough that similar solutions appear repeatedly, but broad enough that each solution has distinctive characteristics. The same logic applies to intelligence. Conway Morris argues that if the tape of life were replayed, intelligence would likely evolve again. Not human intelligence exactly, but something functionally similar. Why? Because intelligence, the ability to process information, learn from experience, solve novel problems, manipulate the environment, provides such enormous survival advantages that evolution keeps moving toward it. We see this in the convergent evolution of complex cognition. Tool use has evolved in primates, elephants, dolphins, corvids, crows and ravens, parrots, and octopuses. Problem-solving abilities appear in species as different as rats and cleaner fish. Social learning, the ability to transmit information between individuals culturally rather than just genetically, has evolved in dozens of lineages. Most striking is the convergent evolution of large, complex brains. Mammals evolved large brains. Birds independently evolved large brains relative to their body size. Cephalopods evolved the largest brains of any invertebrates. 3 completely separate lineages, 3 different brain architectures, but all moving toward greater neural complexity and cognitive capacity. This suggests that intelligence isn't an accident, it's an attractor in evolutionary space. Once nervous systems exist, once sensory organs allow organisms to perceive their environment, once behavior becomes complex enough to benefit from learning and flexibility, there's selection pressure toward greater cognitive ability. The pathway to intelligence is open, and multiple lineages have found it. But, and this is crucial, the intelligence that evolves is always embodied, always shaped by the specific evolutionary history and body plan of the lineage. Octopus intelligence isn't human intelligence. It's distributed across their arms, operates without language, emphasizes visual processing and texture discrimination over abstract reasoning. Corvid intelligence isn't mammalian intelligence. It's built on a different brain architecture, emphasizes spatial memory and social manipulation. Each lineage's intelligence is shaped by its constraints, so Conway Morris is right that intelligence might be inevitable. But the form intelligence takes is contingent on the specific, developmental, anatomical and ecological context of each lineage. Replay the tape, you might get intelligence again. But you wouldn't get humans. You'd get something functionally similar but structurally unique. This synthesis, constrained exploration, resolves the Gould-Conway-Morris debate. Evolution is neither purely contingent nor completely deterministic. It's constrained by physical law, chemical possibility, and developmental architecture, but within those constraints it explores freely. The constraints create repeatability. The freedom creates uniqueness. And the implications are staggering. If convergent evolution operates on Earth, does it operate everywhere? This is the question that keeps astrobiologists awake at night. As we discover more exoplanets, over 5,000 confirmed so far, The question of whether life exists elsewhere becomes less if, and more, what would it look like? And convergent evolution suggests an answer, strikingly similar to life here. The logic is straightforward. Physics is universal. Chemistry is universal. The periodic table is the same on every planet orbiting every star in every galaxy. Water has the same properties everywhere. Carbon forms the same bonds. Light behaves the same way. If these fundamental constraints shape evolution on Earth, channeling it towards certain solutions, then those same constraints should operate on any planet where life arises. Consider vision. On Earth, eyes evolved independently dozens of times. Why? Because light carries information about the environment, and any organism that can detect and interpret that information has an advantage. The physics of light, its wavelength, its speed, how it reflects and refracts, is identical everywhere in the universe. Any alien life on any planet with a transparent atmosphere and a star-providing illumination would face the same problem: how to detect light and extract spatial information from it. According to research in astrobiology published in 2014, convergent evolution might provide a framework for predicting what extraterrestrial life would look like. If the constraints are universal, then the solution should be universal too. Alien eyes might use different chemistry, might have different structures, might be sensitive to different wavelengths, but they'd still be eyes. They'd still use photosensitive molecules, still focus light, still convert photons into neural signals. The same logic applies to locomotion. On any planet with gravity and solid surfaces, organisms will need to move. Legs, fins, wings, or something functionally equivalent. The solutions available are limited by mechanics and energetics. Flying requires generating lift. Swimming requires moving through fluid. Walking requires supporting weight against gravity. The engineering solutions to these problems are constrained by physics. Aliens might have 6 legs instead of 4, or use different materials in their wings, but the basic principles would be recognizable. This extends to intelligence. If intelligence provides survival advantages on Earth, and the independent evolution of complex cognition in mammals, birds, and cephalopods suggests it does, then intelligence should provide similar advantages on other planets. Any environment complex enough to support life, with resources to compete for and dangers to avoid, creates selection pressure toward better information processing, learning, problem solving. The pathway to intelligence may be open wherever life becomes complex enough. Simon Conway Morris took this reasoning to its logical conclusion. In his 2015 book, The Runes of Evolution, he argued that if convergent evolution is universal, then intelligence is probably common in the universe. Not just simple life, complex, cognitive, possibly technological intelligence. The constraints that channel evolution toward intelligence on Earth should operate everywhere. Replay the tape on any habitable planet, and you'll likely get something that thinks. But there's a crucial caveat. The form that intelligence takes would still depend on the specific evolutionary history and environmental context of each planet. Just as octopus intelligence is embodied differently than mammalian intelligence, alien intelligence would be shaped by alien bodies, alien ecologies, alien evolutionary pressures. The function might converge, but the implementation would remain wildly variable. This is why the search for extraterrestrial intelligence focuses on techno-signatures, signs of technology, rather than just biosignatures. Technology is a convergent solution to environmental manipulation. Tool use has evolved independently multiple times on Earth. Any intelligent species that wants to modify its environment beyond what biology alone allows would need technology. The specifics would differ, but certain principles, using external objects as extensions of the body, harnessing energy, manipulating materials, are probably universal. Radio waves are a good example. They propagate through space, carry information efficiently, and can be detected across vast distances. Any technological civilization that discovers electromagnetism, and electromagnetism is a fundamental force, so any civilization studying physics should discover it, might use radio for communication. This is why SETI, the Search for Extraterrestrial Intelligence, has focused on radio signals for decades. Not because aliens would necessarily use radio, but because it's a convergent solution to interstellar communication, given the physics of electromagnetic radiation. Of course, convergent evolution doesn't guarantee identical outcomes. The constraints narrow the solution space, but they don't eliminate all variation. Earth's history demonstrates this. Camera eyes evolved twice, but vertebrate eyes and cephalopod eyes differ in important ways. Vertebrate retinas are wired backward, with blood vessels and nerves in front of photoreceptors, creating a blind spot. Cephalopod retinas are wired correctly, with photoreceptors facing forward. Same function, different implementation. Alien life would show similar patterns: convergence on function, divergence in detail. An alien might have eyes, but those eyes might be sensitive to infrared or ultraviolet rather than visible light, depending on the star they orbit. They might have legs, but the number and arrangement could vary wildly. They might have intelligence, but the cognitive architecture could be radically different from ours, distributed rather than centralized, collective rather than individual, operating on timescales we'd find incomprehensible. Recent work in astrobiology has explored scenarios where life uses different chemistry entirely: silicon-based life instead of carbon-based, methane as a solvent instead of water. According to research from 2024 exploring alternative biochemistries, while carbon and water are optimal for life on Earth, other combinations might work under different planetary conditions. The constraints would differ, which means the evolutionary outcomes would differ. But convergence would still operate within those altered constraint spaces. Life using methane instead of water might still evolve vision, locomotion, intelligence, just implemented through completely different molecular machinery. This brings us to the deepest implication of convergent evolution. The universe might be much less lonely than we thought. The traditional view, following Gould, was that humans are an accident, a cosmic fluke. If you rewound Earth's history and let it play again, intelligence might never evolve. We might get a planet of bacteria, or trilobites, or something completely unrecognizable. Under this view, complex life is rare, intelligent life rarer still, and the universe is probably dominated by sterile rocks and simple microbes. But if Conway Morris is right, if convergent evolution is a universal principle, then intelligence is not an accident. It's a probable outcome of evolution on any planet where life becomes complex enough. The pathways are many, but the destination, something that thinks, that manipulates its environment, that builds technology, is predictable. The Fermi paradox asks, if intelligent life is common, where is everyone? We've been listening for signals for decades and heard nothing. We've searched hundreds of star systems for signs of industrial civilizations and found none. The apparent absence of aliens seems to contradict the idea that intelligence is a common evolutionary outcome. But convergent evolution offers a different perspective. Maybe intelligence is common, but the specific form intelligence takes is incredibly variable. Maybe we're looking for the wrong signals because we're assuming aliens would converge on human-like technologies. Radio telescopes assume aliens use radio. Optical SETI assumes they use lasers. But what if convergent evolution applies to basic biological principles, vision, locomotion, information processing, but not to cultural choices like communication protocols or technological development paths? Or maybe the constraints operate on longer timescales. Life on Earth took 4 billion years to produce technological intelligence. That's most of Earth's habitable history. If that timescale is typical, then even if intelligent life is common, most civilizations haven't reached the technological stage yet, or passed through it long ago and moved on to something we wouldn't recognize as technology. Or maybe convergent evolution has a darker implication. Maybe there are constraints we haven't discovered yet, constraints that limit how long technological civilizations survive. If every intelligent species faces the same challenges, resource depletion, climate instability, the development of destructive technologies, and if the solution space for surviving those challenges is narrow, then maybe convergence isn't just about eyes and wings. Maybe it's about extinction too. This is speculation, of course. We have a sample size of one Earth, one planet, one evolutionary history, one emergence of intelligence. Everything we know about convergent evolution comes from observing independent evolutionary experiments within that single history. We can't yet test whether convergence operates across planets because we haven't found life anywhere else. But the discovery of thousands of exoplanets changes the equation. Many of these planets exist in the habitable zones of their stars, where liquid water could exist on the surface. The James Webb Space Telescope is analysing the atmospheres, searching for biosignatures, oxygen, methane, combinations of gases that would suggest biological activity. Within the next few decades, we might detect signs of life on another world, and when we do, convergent evolution makes a prediction that life, whatever form it takes, will solve fundamental problems in recognisable ways. It will harness energy, it will process information, it will reproduce, compete, adapt. The molecular details might be alien, the specific body plans might be unrecognisable, but the underlying solutions, the ways life solves the universal problems of survival, should show patterns we'd recognise from Earth. Because the constraints aren't accidents, they're features of reality itself: physics, chemistry, the mathematics of development. These aren't parochial to Earth, they're universal. And if the constraints are universal, then the evolutionary outcomes they produce should show universal patterns too. This is what convergent evolution ultimately reveals: that life, while diverse in its implementations, is constrained in its possibilities. The universe isn't a blank canvas where anything imaginable can exist. It's a structured space with rules, with preferred solutions, with attractors that pull evolution towards certain forms regardless of starting conditions. We're not entirely free, but we're not entirely alone either. But understanding convergence doesn't just tell us about aliens. It tells us something profound about ourselves. Humans like to think we're unique, special, the pinnacle of evolution, the only species on Earth with language, culture, technology, abstract thought. And in certain narrow ways, that's true. No other species writes symphonies or builds particle accelerators or contemplates its own existence quite like we do. But convergent evolution suggests that many of the traits we consider uniquely human aren't unique at all. They're convergent solutions that evolution has discovered multiple times, in multiple lineages, in response to similar selection pressures. We're not the endpoint of evolution, we're one implementation of patterns that appear throughout the natural world. Take tool use. For decades, it was considered a defining human characteristic. Then Jane Goodall observed chimpanzees fashioning twigs to fish termites from mounds. Then researchers documented dolphins using sponges to protect their beaks while foraging. Then New Caledonian crows were found crafting hooks from plant material, even manufacturing tools in multiple steps, planning sequences of actions to achieve goals. The list kept growing. Sea otters use rocks to crack open shellfish. Elephants modify branches to swat flies. Octopuses carry coconut shell halves as portable shelters. Tool use isn't a human innovation. It's a convergent solution to the problem of manipulating the environment beyond what anatomy alone allows. Any species with sufficient cognitive capacity and appropriate appendages can discover it. Language seemed more secure as a human hallmark, but the more we study animal communication, the more complex it appears. Dolphins have signature whistles that function like names. Prairie dogs have alarm calls with syntax combining sounds to convey specific information about predators: type, size, colour, speed, trajectory. Humpback whales sing songs that change over time, spreading culturally through populations like musical trends. According to research on vervet monkeys from the 1980s that established foundational principles still studied today, these primates use different alarm calls for different predators: eagles, leopards, snakes, and other vervets respond appropriately to each call without seeing the threat. That's semantic communication. The calls have meaning. They refer to specific things in the external world. That's what we thought language was for. The more researchers look, the more they find communication systems with properties we thought belonged exclusively to human language: reference, syntax, cultural transmission, even creativity. Bees perform waggle dances that communicate the distance and direction to food sources using symbolic representation. The angle of the dance relative to the sun corresponds to the direction. The duration corresponds to the distance. It's abstract, symbolic, learned socially. It's language, just embodied differently. Even consciousness, that most seemingly unique human attribute, shows signs of convergence. Consciousness is notoriously difficult to define, let alone detect in other species. But certain behavioural markers, self-recognition in mirrors, episodic memory, theory of mind, metacognition, suggest that some level of self-awareness exists beyond humans. Great apes recognize themselves in mirrors. So do dolphins. So do elephants. So do magpies. 4 independent evolutionary lineages, 4 different brain structures, but all showing evidence of self-recognition, which most researchers interpret as a marker of self-awareness. Scrub jays demonstrate episodic memory. They remember not just where they cached food, but what they cached and when, and they adjust their behaviour based on whether the food would have spoiled. That's mental time travel. That's experiencing events as moments in personal history rather than just responding to present stimuli. It's a cognitive capacity we thought required human-level intelligence. Dolphins and great apes show theory of mind, understanding that other individuals have mental states different from their own. They can anticipate what others know or don't know, what others intend, what others will do. That's a sophisticated cognitive achievement, requiring models of other minds within your own mind. Even corvids, crows and ravens, demonstrate planning abilities that rival primates. They can solve multi-step problems, use tools in sequence, delay gratification, and even engage in what looks like play. New Caledonian crows can solve 8-step puzzles, requiring them to use one tool to obtain another tool, to obtain a third tool that finally gets them the reward. That's causal reasoning. That's understanding means-ends relationships across multiple levels of abstraction. All of this points to the same conclusion. The cognitive capacities we thought made us unique are actually convergent solutions to the problems of surviving in complex social and ecological environments. Intelligence, defined broadly as flexible problem-solving, learning, planning, social cognition, has evolved independently in multiple lineages because it's advantageous. The specific implementations differ, but the underlying functions converge. This realization is humbling. We're not the only intelligent species on Earth. We're not even the only species with culture, language, tool use, self-awareness, theory of mind, or episodic memory. We're one species among many that has stumbled onto these solutions. What makes us distinctive isn't that we have these capacities, but how we combine them and to what extent we've developed them. Our particular implementation of intelligence emphasizes abstract symbolic reasoning, cumulative culture, and technological innovation to an unusual degree. But those aren't categorically different from what other species do. They're quantitative differences, not qualitative ones. We're further along certain cognitive dimensions, but we're walking paths that other species have discovered too. This has implications for how we think about ourselves and our place in nature. If intelligence is convergent, if consciousness appears in multiple lineages, if language and tool use and culture aren't uniquely human, then the sharp line we've drawn between humans and other animals starts to blur. We're not separate from nature. We're a product of the same evolutionary processes, subject to the same constraints, discovering the same solutions. Our intelligence, our consciousness, our sense of self, none of it arose in isolation. It's all part of a broader pattern of convergence toward complexity that runs through multiple branches of the tree of life. But there's another implication, darker and more urgent. If human cognitive traits are convergent, if they've evolved multiple times independently, then they can also be lost. Evolution isn't directional. It doesn't inevitably move toward greater complexity or intelligence. It moves toward whatever provides reproductive advantage in the current environment. And if environmental conditions change such that intelligence becomes costly rather than beneficial, selection will favour its reduction. We see this in evolutionary history. Many lineages have lost complexity. Cave fish lost their eyes because maintaining eyes in perpetual darkness is energetically expensive with no benefit. Parasites often lose complex organ systems because their hosts provide everything they need. Island species frequently lose flight because without predators, the energy cost of maintaining flight muscles exceeds the benefit. Intelligence is expensive. The human brain consumes 20% of our metabolic energy, despite being only 2% of body mass. Maintaining large brains, raising offspring with extended developmental periods, supporting the social structures that make cumulative culture possible, all of this requires resources. If those resources become scarce, or if environmental conditions change such that simpler cognitive strategies work just as well, evolution could easily move away from intelligence. This is the constraint we don't like to acknowledge. evolution discovered intelligence because intelligence was advantageous in certain ecological and social contexts. But if those contexts change, intelligence might become a liability. And unlike the physical constraints that make convergent evolution predictable, gravity, chemistry, development, the selective pressures that favour intelligence are contingent on environmental conditions. Climate change, habitat destruction, resource depletion, pollution. All of these alter the selective landscape. They change what's advantageous, what survives, what reproduces. If we make the planet inhospitable enough to complex life, evolution will favour simplicity. The cognitive capacities we're so proud of could be selected against in a future we're actively creating. The bacteria would be fine. They've survived every mass extinction in Earth's history. Simple, adaptable, capable of exploiting nearly any environment. But complex multicellular organisms, organisms with expensive brains and slow reproductive cycles, and specialised ecological requirements, they're vulnerable. We're vulnerable. Convergent evolution tells us that intelligence can evolve, but it doesn't guarantee that intelligence, once evolved, will persist. The same constraints that channel evolution towards certain solutions can channel it away from those solutions if conditions change. We are ultimately subject to the same evolutionary forces as every other organism on Earth. We're not exempt from natural selection just because we're clever enough to understand it. This is the final lesson of convergent evolution. We're both inevitable and fragile. Inevitable in the sense that given the right conditions, complex environments, social competition, ecological pressures, intelligence will likely emerge. But fragile in the sense that those conditions can change, and when they do, there's no guarantee that intelligence remains advantageous enough to persist. The pathways to intelligence are many, but they can be closed. Not by meteor strikes or supervolcanoes, though those would certainly work, by slower processes, by degrading the ecosystems that support complex life, by simplifying the world until simple solutions outcompete complex ones. We think of ourselves as the pinnacle of evolution, the endpoint of a long journey from simple to complex. But evolution has no endpoint. It's a process, not a progress, and processes can run in either direction. Yet within these constraints lies a paradox. The same forces that limit what evolution can produce also enable novelty. This seems contradictory. How can constraints, limitations on possibility, create new possibilities? But it happens constantly, and understanding how reveals something fundamental about how evolution actually works. Constraints don't just eliminate options, they create structure, and structure creates the possibility of modular innovation. When certain features become fixed, locked in by developmental, physical, or functional constraints, they become platforms upon which new variations can be built. The constraint provides stability. The stability enables exploration. Consider body segmentation. Once arthropods evolved a segmented body plan, that constraint became a foundation. You can't easily remove segmentation from an arthropod without destroying the developmental program. It's locked in, constrained. But within that constraint, evolution can modify individual segments, specialize them for different functions, turn some into walking legs, some into mouth parts, some into reproductive structures, some into sensory antennae. The constraint, segmentation itself, enables the innovation, segment specialization. Without the fixed structure, you couldn't have the modular variation. This is how constraints create opportunity. According to research published in 2023 on evolutionary innovation, the most significant evolutionary novelties often arise not by abandoning existing constraints, but by redeploying conserved developmental mechanisms in new contexts. The genetic toolkit is constrained. Most animals use the same basic developmental genes, the same regulatory networks, but those conserved tools can be deployed in new patterns, new locations, new timing. Beetle horns are a classic example. According to research on developmental evolution by Armin Motschek, horns in beetles evolved by redeploying leg development genes in new locations on the head and thorax. The genes themselves are ancient, conserved across arthropods. The constraint is that you have this particular genetic toolkit for building appendages. The innovation is using those tools to build something that isn't a leg at all. This pattern repeats throughout evolution. Feathers evolved from scales using modified versions of the same developmental pathways that build reptile scales. Eyes evolved by redeploying genes that originally patterned other body structures. The inner ear bones of mammals evolved from the jaw bones of ancestral reptiles, repurposed through changes in developmental timing and location. Evolution isn't creating new genetic programs from scratch, it's tinkering with existing ones, constrained by what's already there, but innovative in how those constrained elements get recombined, redeployed, modified. This is why evolutionary novelty often appears as a mosaic, new features built from old parts. The constraints determine which parts are available. The innovation comes from arranging those parts in new configurations. Research from 2024 exploring morphological innovation in skink lizards found that most evolutionary change occurs through what they call elaboration, gradual variation along established axes of morphospace. But periodically, evolutionary bursts occur, rapid innovations that open new regions of morphospace. These bursts aren't random. They happen when constraints are temporarily relaxed, or when existing constraints interact in new ways to create novel possibilities. The turtle shell is another case. According to 2024 research, the shell imposes severe constraints on turtle evolution. It limits how body proportions can change, how limbs can modify, even how breathing mechanics can evolve. But the shell itself was an innovation, arising through redeployment of rib development pathways. Once established, it became a constraint that channeled subsequent evolution. Turtles have remained remarkably conservative morphologically for 200 million years because the shell constrains so much. Yet within those constraints, turtles have diversified into hundreds of species exploiting different ecologies. This interplay between constraint and innovation is the engine of evolutionary novelty. Constraints narrow the search space. Innovation explores within that narrowed space. When innovations become fixed, they create new constraints, which enable new forms of innovation. It's recursive. Each layer of constraint creates opportunities for the next layer of innovation, which becomes the constraint for the layer after that. This is how complexity builds over evolutionary time. Not by escaping constraints, but by building on them. And this brings us back to the deepest insight of convergent evolution. The patterns we see, crabs evolving 5 times, eyes evolving dozens of times, intelligence evolving independently in multiple lineages, aren't accidents. They're the signature of a structured search through possibility space. Evolution isn't randomly wandering through infinite options. It's following gradients, flowing through valleys, converging on attractors that exist because of the fundamental structure of physical reality. This structure makes evolution partially predictable. Not completely. There's still contingency, still randomness, still path dependence, but partially. Given similar starting conditions and similar selection pressures, evolution will tend to discover similar solutions because those solutions are the ones that actually work within the constraints. This has profound implications for how we understand life. Life isn't infinitely malleable. It's constrained by physics, by chemistry, by development, by history. Those constraints mean that the space of possible organisms is far smaller than the space of imaginable organisms. Most science fiction aliens couldn't exist, not because we lack imagination, but because the constraints that govern how matter self-organizes into living systems eliminate most possibilities before they can be realized. But within the constraints, within the narrow channels that physics and chemistry permit, within the viable regions of morphospace, life is exuberantly diverse. The constraints don't prevent variation, they guide it, channel it, make it productive rather than random. This is why Earth's biosphere, despite 3.5 billion years of evolution, despite mass extinctions and climate shifts and continental drift, despite an incomprehensible number of genetic mutations and environmental pressures, has produced only about 35 basic animal body plans. The constraints winnowed possibilities down to a small set of viable architectures. But within those architectures, we get millions of species, each uniquely adapted to its ecological niche, each a variation on constrained themes. We started by asking why life keeps solving the same problems the same way. The answer isn't that evolution is predetermined or that there's some cosmic blueprint. The answer is structure. Reality has structure, physics has structure, chemistry has structure, development has structure. and that structure constrains what's possible, channels what's probable, creates attractors toward which evolution converges. The same eyes, the same wings, the same crabs, not because evolution remembers previous solutions, but because the landscape of possibility has valleys, and water always flows downhill. This doesn't diminish the wonder of life. If anything, it deepens it. Because it means that the patterns we observe, the convergences, the repeatability, the apparent inevitability of certain solutions aren't illusions or coincidences. They're revelations of something deeper, the hidden rules, the invisible architecture, the structure of the possible itself. Evolution explores, but it explores a landscape that already exists, shaped by laws we're only beginning to understand. The journey is contingent, unpredictable in its details, unique in its specifics. But the destinations, the regions of possibility that actually work, that can be built, that can survive, those are constrained, limited, discoverable multiple times because they exist independent of any particular evolutionary path. This is what convergent evolution teaches us. That beneath the chaos of mutation and selection, beneath the accidents of history and the randomness of genetic drift, there are patterns, structures, rules, not imposed from above but emerging from below, from the fundamental properties of matter and energy and information. Life isn't free to be anything. But within its constraints, within the narrow regions of possibility that the universe permits, it finds ways to thrive that are both predictable and endlessly creative. The same solutions discovered independently again and again, each time unique in implementation but recognizable in function. This is the mystery and the revelation, that we can predict what evolution will discover because the discoveries are already there, waiting in the structure of the possible. But we cannot predict the exact path, the specific details, the unique beauty of each solution. The tape of life, if replayed, would produce similar forms, similar eyes, similar wings, similar intelligence, because the constraints that channel evolution are universal. But each replay would be beautiful in its own way, unique in its particulars, a fresh exploration of ancient possibilities. We are all of us, every organism on this planet, every potential organism on distant worlds, explorers of the same landscape, the landscape of what's possible given the laws of physics, the rules of chemistry, the logic of development. We walk different paths, but we converge on similar summits, not because the journey was predetermined, but because the mountains were always there, waiting to be climbed. This is convergent evolution, not proof of design, but evidence of structure. Not cosmic programming, but physical law. Not fate, but constraints that make certain destinations inevitable, given enough time, enough variation, enough selection. The hidden rules constraining all living things, revealed through the pattern of their solutions, repeated across lineages, across continents, across deep time, telling us that life, while wildly creative in its details, is following a grammar we're only beginning to read.